depends on whether the traits are controlled by genes on autosomes or by genes of sex chromosomes. Now, in this unit, we will focus on the works of Gregor Mendel in the field of genetics. Important terms in genetics will also be discussed together with the laws of inheritance and apply the principles of probability and statistics in solving genetic problems using Punnett square. Important terminologies in Mendelian inheritance. Trait. Trait is any characteristics that can be passed from parent to offspring. Next is alleles. Alleles are two genes that occupy the same position on homologous chromosomes and that cover the same trait, that this can be dominant or recessive. Next, genotype. Genotype is the genetic makeup of an organism, gene combination for a trait. Next is phenotype. Phenotype is the physical appearance of an organism, the physical features resulting from a genotype. For example is red and white. Next, genotype. Genotype is the genetic makeup of an organism, gene combination for a trait. Next is phenotype. Phenotype is the physical appearance of an organism, the physical features resulting from a genotype. For example is red and white. Next, genotype. Genotype is the genetic makeup of an organism, gene combination for a trait. Next is phenotype. Phenotype is the physical appearance of an organism, the physical features resulting from a genotype. For example is red and white. Next, homozygous. Homozygous having identical genes, one from each parent, for a particular characteristics. Gene combination involving two dominant or two recessive genes, also called pure. Next, heterozygous, having two different genes for a particular characteristics. Gene combination, one dominant and one recessive allele, also called hybrid. Okay, monohybrid, a genetic cross involving a single or pair of gene or one trait. Parents differ by single trait, for example, flower color. Next is P or the parental generation. F1 refers to first filial generation, offspring from a genetic cross. F2 also is a second filial generation of a genetic cross. Finally, dihybrid is a genetic cross involving two pairs of gene or two traits. For example, flower color and plant height. Finally, dihybrid is a genetic cross involving two pairs of gene or two traits. For example, flower color and plant height. Law of inheritance. There is three types of law of inheritance. These are the law of segregation, second law of dominance, and third law of independent assortment. So the first one is law of segregation. According to the law of segregation, only one of two gene copies present in an organism is distributed to each gamete that it makes, and the location of the gene copy is random. When an egg and sperm unite in fertilization, they form a new organism whose genotype consists of the alleles contained in the gametes. Next is law of dominance. Law of dominance states that when two alleles of an inherited pair is heterozygous, then the allele that is expressed is dominant, whereas the allele that is not expressed is recessive. Let's proceed to the law of independent assortment. Independent assortment states that the alleles of two or more different genes get into gametes independently of one another. In other words, the allele agamete receives for one gene does not influence the allele receive or for one another gene. This law can be illustrated using dihybrid process. Punnett square. Reynold Punnett and William Bates were among the first English geneticists. Punnett devised the Punnett square to depict the number and variety of genetic combinations and had a role in shaping the Hardy-Weinberg law. Punnett and Bateson co-discovered coupling or gene linkage. William Bateson brought Mendel's laws to the attention of English scientists. So, how to compute in a Punnett square? Steps in using the Punnett square. Step 1. Assign symbol for each allele. So, let's say that trait is T is height of the plant. Uppercase letter T is tall and smaller is dwarf. Always remember, use uppercase letter for the dominant allele and lowercase letter for the recessive allele. Step 2. Determine the genotypes of each plant. Double big letter T is pure bred tall and double small letter T is pure bred dwarf. Tips. Read the problem carefully. Look for clues to identify if the parent is homozygous or heterozygous. Third step. Determine the kind of gametes produced by each parent. Remember, apply Mendel's principles of segregation. Step number 4, it's time for the Punnett square. Fill in each grid with 2 alleles and 1 from the parent along the top of 1 from the parent along the side. The letters in the middle show you all possible combinations of alleles that can happen when these gametes meeting during fertilization. Next is determine the phenotype of each offspring. Remember to obey Mendel's principle of dominance. Step 5. Identify the genotypic and phenotypic ratio. If genotypic ratio, write all the possible genotypes and count its presence in the Punnett square. For phenotypic ratio, write all the possible phenotypes and count its presence in the Punnett square. For example, in genotypic, there is double T and big T, or uppercase T and lowercase t is equal to 4, and small letter t is equal to 0. And for the phenotypic ratio, all tall for 100% is tall. There is all T in the Punnett square. So the last step, step 6, give a specific answer the given questions from the problem.